Okay, circulatory system part two. So in the second part here, we're going to talk about further about some specific heart structures. So we kind of talked about the basic parts of the heart in the previous lessons. So how we have the top chambers, the atria, the bottom chambers, the ventricles, how we have that septum dividing the left and the right side of the heart. We talked about the vena cava, which brings blood back to the heart from the top and from the bottom of our bodies. And we talked about the aorta, which takes that oxygenated blood out and to the rest of the body. So we kind of talked about a few of our basics already. And so the next step that we're going to talk about is our valves. This little animation is very distracting, but it's actually a good little animation to show us how the valves work. So there's basically two types of valves in your heart. So there's the AV valves, or the atrioventricular valves, and then there's the semilunar valves. So on this first slide, we're going to talk about the atrioventricular valves. Now, the easiest way to remember, distinguish between the AV valves and the semilunar valves is that if you think about it, the atrioventricular valves are located between the atria and the ventricles. So if you think AV, think atria, ventricles, then that kind of helps you remember where those valves are located. So the atrioventricular valves actually separate the atria from the bottom chambers from the ventricles. Now in this animation here, it's kind of weird because sometimes kids look at this and they're like, I don't even see the atria. All I see is the ventricles. Well, when we do our heart dissection, you're going to see that in fact, this weird kind of flap of tissue here on both sides, you see how there's this kind of meaty kind of piece of tissue there that's kind of contracting, that's actually the atria, okay? And they're quite thin-walled and quite small in comparison to the ventricles. So these big things that we see at the bottom here, those, of course, those big chambers are the ventricles. Now, in terms of which side's which, keep in mind that here's that vena cava here, right? So that means that this is the right side of the heart, and then over here we have the left side of the heart. Okay, so we have two AV valves. So we have one on the right side of our heart and we have one on the left side of our heart. So if we take a look at the valve here, if you look at this valve here that I'm kind of indicating, right, this one here, you can see that it has three flaps. So that actually is our tricuspid valve. So on the right side of our heart, we have a three flap valve called the tricuspid, which helps control flow from the atria to the ventricles in terms of blood. Now, if we go to the left side of our heart here, okay, this one is our bicuspid, or our mitral valve, and that is a two-flap valve, and that one actually separates the left atria from the left ventricle and helps control that blood flow. Okay, so just keep in mind that the mitral valve, in our program of studies, we call it the bicuspid. Okay, so tricuspid on the right side, three flaps, bicuspid on the left side, two flaps. Now, the other thing that you may notice that you have here is you have these kind of like parachute kind of strings here. Do you see that? Kind of little like parachute, kind of, they look like they're holding on to the valves. Well, those are called the chordae tendineae. And chordae tendineae are important because what they actually do is they're very flexible, very strong little strings of tissue that actually help the valves pull closed and remain taut. Okay, because if the chordae tendineae weren't there, then the valves would kind of just be kind of floppy, and then you'd get backflow, blood flowing back from the ventricles back into the atria every time your heart actually contracted. So those chordae tendineae help maintain the tautness of the flaps of the valves and help ensure that they close very accurately and very tightly. Okay. Now, if we look at our next valve here, we have, or our next category of valves, we have the semilunar valves. Okay, the semilunar valves are the valves that actually control blood going out and into the heart. Okay, so our cuspid valves, like we looked at last time, our tricuspid and our bicuspid valve, those ones control blood flow within the heart, right? So between the atria and the ventricles. The semilunar valves, they control blood that's going to be moving into and out of the heart, all right? So if we take a look on our diagram here, we have two semilunar valves, and the first one is the pulmonary valve. And I want you to remember from our previous lesson that the pulmonary valve is associated with the lungs, right? So anything that has that term pulmonary, we know that that has something to do with the lungs. So remember we talked about pulmonary artery, pulmonary vein, now we have a pulmonary valve. So the pulmonary valve actually allows blood to leave that right ventricle and go back to the lungs. Okay, so you see this valve right here. This valve actually controls blood flow out of the right side of the heart, and then that blood is going to go back to our lungs to pick up more oxygen. Okay, and so it helps control that blood flow. Now the other one is called the aortic valve, and you can see it kind of tucked underneath the pulmonary valve. And again, think of the name. So aortic valve, right? Obviously, think of aorta. And so the aortic valve helps promote blood flow out of that left side of the heart and into that aorta so that it can be distributed through the rest of the body. Okay, so AV valves within the heart, separating atria and ventricles. Semilunar valves, the pulmonary and the aortic, controlling blood flow to the lungs and into the aorta, respectively. So if we look at our little animation here, circulation within the heart is one of the more important concepts in terms of the program of study. So you, in a test situation, need to be able to start at any point in the heart and then be able to understand which direction the blood is going to flow. So this little animation kind of helps us with that. So when the text is highlighted in green, then that shows like where the blood currently is, and then the next one that is highlighted green shows where it goes from there. Okay, so if we start at the right atrium, then we go through the tricuspid into the right ventricle, through the pulmonary valve, into the pulmonary artery, pulmonary vein, left atrium, bicuspid, left ventricle, aortic valve, into the aorta, and then out to the rest of our body. Okay, so we're going to do a couple activities to help with that, but you definitely need to know the direction of that blood flow. Okay, so this slide is important for that. Now, in our program of studies, there also is an additional kind of terminology when it comes to the difference between the pulmonary circulatory system and what we call the systemic circulatory system. So I'm sure you probably already can tell me a little bit about what the pulmonary part of the circulatory system is. And the pulmonary part has to do with any part of the lung that, or any part of the heart rather, that is responsible for taking blood to and from the lungs. Okay, so that would be our pulmonary circulatory system. So when you think of like your pulmonary artery, your pulmonary vein, okay, those are obviously going to be part of your pulmonary circulatory system. Now, on the other hand, vessels that carry blood to and from the body's other tissues are part of our systemic. Okay, so if we look at our aorta, for example, okay, that's going to be part of our systemic circulatory system because that's delivering blood to subsidiary arteries and then eventually to the rest of our tissues. So pulmonary is all about the vessels that carry blood to and from the lungs. Systemic is anything else, okay? Anytime it isn't associated with moving blood to or from the lungs specifically, that's going to be part of our systemic circulatory system. So we already talked a little bit about the capillaries in our previous lesson in terms of structure, right? So we talked about that they have a very small lumen. They're like the size of a human hair. Only one red blood cell can go through at a time because they're super, super fine. We talked about the fact that another reason they're really fine is because they have to work their way in and around all of our tissues. Every cell in our body has to have access to a capillary in order for the nutrient and gas and waste exchange, okay? Because that is the job of the capillary. It's to allow the exchange of nutrients, oxygen, and wastes between body cells and the circulatory system. So there's some terminology that we should talk about when it comes to plasma and the difference between plasma and extracellular fluid and lymphatic fluid. Okay, so compositionally, plasma and extracellular fluid, or ECF, okay, so ECF stands for extracellular fluid. Extracellular fluid, plasma, and lymph, which is lymphatic fluid, they all are very, very similar in terms of composition. So they're mostly water, but they will also have like some proteins in there and some other things too, which we'll talk about a little bit later. But what the difference is between them is, where are these fluids? Okay, so let's talk about that for a minute. So plasma is the term that we use for that water-based fluid when it is found in the circulatory system. Okay, so in the veins, in the arteries, in the capillaries. Okay, that's plasma. Okay, because you think of, like, when we think of plasma, we always think of blood, right? So when it is in our vessels, our blood vessels, then we call it plasma. Now, when it is bathing the cells that make up our tissues, then it's called extracellular fluid. So if you kind of just think of any tissue, so say you have like muscle tissue or liver tissue or whatever. So all tissues are made up of a whole bunch of cells, right, together. And then just going to switch colors here. And then the ECF, or the extracellular fluid, is bathing those cells. Okay, so your cells aren't just like, rolling around, clanking around in your body. They're bathed by that extracellular fluid, okay? So when it's outside of the circulatory system and bathing the cells, then we call it extracellular fluid. Now, there's one other term that I mentioned too, which is lymph. Now, we're going to talk about the lymphatic system a little bit later, but this fluid, okay, this water-based plasma-like fluid, when it is in the lymphatic vessels or the lymph nodes, okay, then we call it lymph. And that one's usually easy for kids to remember because they're like, okay, well, lymphatic system, lymph, I get it. So if it's in the lymphatic vessels or the lymph nodes, then we call it lymph. So plasma in the circulatory vessels, ECF, or extracellular fluid, bathing our cells, so not inside those circulatory system vessels, and lymph when it's in our lymphatic vessels and our lymph nodes, okay? So the fluid that enters the tissue from the bloodstream is rich in oxygen and nutrients, including things like glucose, amino acids, etc., okay, because those things are going to be transported to our cells for not only cellular respiration, but other cellular processes. So this is a very important diagram in terms of understanding what's happening here and kind of going back to our circulatory system vessels. So when we look at our circulatory system vessels, I want you to remember that blue always stands for deoxygenated, and red in these diagrams stands for oxygenated blood. So we have oxygen-rich blood on the arterial side here, and we have oxygen-poor blood on the vein side there, right? So if we kind of continued this, because you'll notice that we kind of are going like from an artery, which remember is the larger type of vessel, and then narrowing into the arteriole. So if we kind of backed up all the way to the largest artery here, where would we be, kiddos? Do you remember? We would be at the aorta, right? Because that is the largest of our arteries. So think of it this way. So your heart pumps, left ventricle pushes blood into the aorta, and eventually the blood from the aorta ends up in our arteries and then in our arterioles, and then eventually will go into this capillary bed. And the capillary bed always kind of looks like a fishnet. So you can think of it like that. Anytime you see kind of this fishnet sort of structure, that's going to be a capillary bed, and that's where nutrient and gas exchange happens, because that's the whole purpose of the capillaries, right? Then if we go to the other side here, we have a vein. And then notice that the vein narrows down into the smaller version of that vessel, which is the venule. And so if we backed up, where would we kind of be there, kiddos? What's our largest vein? If you think of what our largest vein is, remember that's going to be our vena cava. So if we back up there, we're going to have our superior and our inferior vena cava. Okay, kiddos, so what's happening here then, generally speaking? So I'm just going to change back to green. Okay, so what's happening here then is blood is coming from the aorta, it's flowing into our arteries, and then eventually it's going into the arterioles. Now, notice that you have little sphincters here, right? And remember, the sphincters in our capillary beds are the same as other sphincters, in the sense that they control fluid flow and make sure that fluid doesn't flow too quickly. So those little sphincters kind of allow the blood pressure to kind of be reduced a little bit, because remember the capillaries are super fine. They can burst really easily. So we want to be able to regulate that blood pressure. Okay, so what's happening here? As the blood flows into the capillary bed, you can notice that it kind of then turns purpley here, right? And that tells us that as we move along this capillary bed, oxygen and nutrients are moving from the capillaries into the surrounding tissue. Because remember, this isn't just like capillaries lost in space here. These capillaries are embedded in your tissues, right? So you have all your cells, right, kind of embedded, and the capillaries are wrapped around those cells. And so oxygen is going to diffuse, right, through those pores in the capillary to the cells, and nutrients are going to do the same. So that's why as you move along here, we end up with it looking like purpley, because it's becoming deoxygenated as the blood moves through that capillary bed. Now, I also want you to think about what's going to move in. So if we have oxygen and nutrients moving from the capillaries into the cells, what's going to be leaving the cells and moving in to the capillaries? So oxygen, nutrients move in, and what's going to move out, or move in rather, to the capillaries? We're going to have waste products, CO2, moving in, okay? So oxygen, nutrients leaving the capillaries and going to those cells, and then the cells are getting rid of CO2 and waste products and putting those into the capillary bed so they can be dealt with by our circulatory and other systems like the respiratory system. So by the time we get to the venule end over here and to the vein end and eventually to the vena cava, the blood is now low oxygen, low nutrient, but high waste, high CO2 and high waste. Okay? Now, in terms of terminology, there's two terms that are important with respect to these diagrams of this type. So filtration, which is the movement of fluids and their dissolved contents out of the capillaries, and then absorption, which is the movement of fluids and their dissolved contents into the capillaries. So in terms of filtration, we're going to have oxygen and nutrients, right, being filtered in out of the capillaries and into the surrounding tissues. And then we're going to have CO2 and waste products being absorbed back into the capillaries so we can get rid of them eventually. So this is just another version of the same type of diagram here, just a different look. So again, you can see this one's kind of cool, I like, because it shows the difference between like the veins and the venules and so on. So remember, our veins have that large lumen, they have that thinner wall. We don't see the valves here because they're just not relevant to this particular diagram. And then you have venules, which are smaller versions of the same thing. So little thinner wall, still a big lumen, etc. And then on the other side, we have our arteries, right? And remember, our arteries have our narrow lumen, thicker muscular wall, which is capable of constricting, dilating, and then we have our smaller version, which is the arterioles, okay? So this is kind of what I talked about on the previous slide, how we have our cells that make up tissues, and then the capillary bed, right, which is this part here. The capillary bed is going to be supplying those cells with oxygen and nutrients, and then getting rid of the waste products. So remember, this part here is going to be the filtration, right? So we have oxygen and nutrients moving out of the capillary bed and into the cells and tissues. And then this part here is going to be the absorption, right? So that's the part where the cells are going to get rid of CO2 and waste products. And then the CO2 and the waste products are going to go into the capillaries and then out the venule end so that we can eventually get rid of them, okay? Whether that's by exhaling or shunting it back into the lymphatic system or whatever, but we'll talk about that a little bit later anyway. Like the previous diagram too, you can see that the blood that's coming from the heart into the capillary bed is red, okay, that nice bright pinky red color. So high oxygen, high nutrients, but then by the time we get to the venule and the vein end here, we have low oxygen, low nutrients, but we have high waste. So when it comes to capillary fluid exchange, there's a couple terms here that are important, and we're just going to talk a little bit more about what kinds of things can capillaries actually move in and out, and what kinds of things are not supposed to move in and out of our circulatory system, okay? So small substances like oxygen for cellular respiration and CO2, which is the waste product of cellular respiration, can move fairly easily through those capillaries, and it just is a natural concentration gradient, right? Moving from high concentration to low concentration. When it comes to things that are a little bit larger, okay, like sugars and salts and urea, and remember, urea is the less toxic form of ammonia, right? So your liver will convert ammonia to urea because it's not as harmful as ammonia itself. And so we have hydrostatic pressure, which is the pressure that's pushing against the wall of the capillaries, okay? Because remember, that blood's coming in, there's blood pressure there, you get that pressure of that fluid pushing, and then that's what helps the materials like the sugars and the salts, etc., push out of that capillary and into the surrounding tissues so that we have nutrients for all those cell processes that we need. So this diagram does quite a good job really of showing the things that can move out and the things that have to stay in. So once again, at the arterial end, which is the blood coming from the left ventricle, remember this blood here is going to be high oxygen and it's going to be high nutrient. So that's why we have this nice bright red color here. And then if we go to the other side, which is the venous end, or the end where the vein is, this blood is going back to the heart, so going to end up in either superior or inferior vena cava, go back to that right side of the heart so that it can go and pick up more oxygen. And then you'll notice we have the blue here, which means that we now have low oxygen, we have low nutrient, and we have high waste, okay? So anywhere you see a little green arrow, that's refers to osmotic pressure, and anywhere where you see the little yellow arrow, that refers to blood pressure. And remember, blood pressure is the pressure of the blood as it's moving through the vessel, okay? So let's take a look here and see what kinds of things can move in and what kinds of things can move out. So I want you to notice that at the beginning here of the process, because you can think of the blood is flowing this way, right? Okay, because we're going from oxygenated to deoxygenated. So notice that you have water moving out, okay? And notice that you have oxygen moving out. Notice that you have amino acids and glucose moving out, because those are all things that the cells that are around here, right, are going to need for cellular respiration and metabolic cell processes, right? And then one thing I want you to notice here too is as we get closer to the venous end, this is where you start to have CO2 moving in and other waste products like the urea that we talked about moving in, okay? Now, the other thing I want you to notice when we look at what's going through here, okay, is that there's certain things that can't get pushed out of the capillaries because they're too big and they need to stay in the blood. So that's going to be things like red blood cells. That's going to be things like plasma proteins. So plasma proteins are not amino acids, okay? Plasma proteins are things like albumins, which need to stay in the bloodstream, okay? So they shouldn't be pushed out through the capillary bed. So anything that is, and so then we have like platelets and white blood cells. So anything that is too large to fit through those pores of the capillary is not going to move, okay? Those things are going to stay in the bloodstream.